Hello. Okay. Um, so today we're welcoming our speaker, Richard Roy, who is from originally around Toronto. He started his university studies in Melbourne, then finished back in Quebec, and then did his graduate studies at Université Laval, also in Quebec, before moving to France to do his postdoc with Jean-Marc Algy and Pierre Champon, thank you, at the Institute of Genetics, Molecular and Cellular Biology in Strasbourg, France, studying transcription. Then he moved to Dartmouth, where Victor Ambros was at the time, for another postdoc experience studying the cell cycle. And after that, started his own lab at McGill, where one of the focuses of his lab is to study how metabolic signaling coordinates cellular functions, often non-cell autonomously, including cell division in the germline. And to do this, they use the nematode C. elegans, and they capitalize on an unusual and strange state that C. elegans can enter called the Dower state, where the animals don't eat at all, live many times longer than they normally would, and continue to move around and behave. And then after they come out of it, they have a normal lifespan. So very impressive. And I'm excited to hear what he's going to tell us today. So thank you for coming. Oops.
Well, first of all, thanks very much, Claire, for inviting me to come to this wonderful university. And I want to say that I've had just extraordinary interactions with everybody today. I learned so much. So thanks for taking the time. Thanks to students for coming to lunch with me. And hopefully I'll be able to share some of the new, what I think, of course, but I'm biased, some very exciting data that we've more or less developed over the last 3 years and 4 years, maybe longer actually, but largely based on genetic analysis. And of course, coming from the laboratory that I came from, Victor's always the first guy to say how important it is to use genetic analysis to really frame your ideas and your research program, because it's very unlikely that you'll be disappointed. And so I'd like to start by saying that probably for the last decade and a bit, my lab's been really interested in stem cells. And I don't think I'm alone in this room in sharing this interest in this wonderful cell type. And I think that it's justified because stem cells do these extraordinary things. And they're extraordinary, particularly from the perspective of cell biologists, and they're extraordinary because of their clinical potential. And this great, extraordinary aspect of stem cell biology is all dependent on the way that these stem cells actually divide. And it's because they have this property of self-renewing that allows these stem cells to generate specific cell types that can contribute to tissues, et cetera, but also retaining the ability to go back to the mother stem cell phenotype. So from a cell biological point of view, the cell divisions that these stem cells undergo are particularly interesting because they're regulated in a very interesting manner. The cell divisions that stem cells can undergo can be asymmetric, and those asymmetric divisions might be regulated by cell intrinsic properties or factors, or they can also be regulated by the environment, those cells that are surrounding the stem cells. But they can also be symmetric. And sometimes stem cells can be symmetrically dividing or asymmetrically dividing. So the divisions really get us excited because it's the way that they divide and this ability to retain the self-renewal potential that actually make them so important. But the strange thing is, and I think from my perspective in my laboratory, is that stem cells, despite the fact that they do these extraordinary divisions, largely don't like to divide. In fact, a lot of stem cells would just rather stay in a quiescent state. a non-dividing state within their environment, their niche. And it's only under certain circumstances that signals will impinge on those cells to tell them or instruct them to start to undergo divisions. But otherwise, they would remain in an arrested state until they get these particular cues. And we can argue up and down why stem cells might do that. But I think the most common view is that they're really trying to protect the genetic information. And every time you divide, you actually subject yourself to some kinds of errors. So we've been very interested in how these stem cells can retain this ability to stay in an arrested state for long periods of time. And as Claire said, we turn to C. elegans and the power of genetic analysis in order to understand the genes and how those genes function to ensure that those stem cells can stay in this quiescent state, even in situations where they're in abundance. And the stem cell populations in C. elegans are not very numerous. There's two really clear stem cell lineages that people look at. Victor's making great headway with understanding how the timing of a hypodermal stem cell-like lineage is being regulated. But the germ cells, which is something that's very, I think, a prominent figure of many of the biologists here, the germ cells also stem from a stem cell population that's born very early during embryogenesis. And I try to show that here in this cartoon where the entire germ cell population, or the germ line, arises from two cells that are born quite early during embryogenesis and remain in a non-dividing state. And we can refer to these as Z2, Z3, or for you, Z2, Z3. And once as animals leave embryogenesis or emerge from the hatch and start feeding, Z2, Z3 will start to divide. And they'll divide in an uninterrupted manner to populate two gonad arms in this animal and to give rise to all the gametes that'll eventually fuse together and give rise to the animals in the next generation. And as I said, these divisions are ongoing and uninterrupted under normal circumstances. But under some conditions, those divisions can be arrested. And I'll just give you a quick primer on C. elegans development, shown here, which is basically after embryogenesis, they'll go through 4 larval stages. And just kind of in the end of the first larval stage, the animal assesses its environment and has to make a decision whether it's going to continue through normal reproductive development, or if growth conditions are suboptimal, it can opt out of reproductive development and enter, as Claire mentioned earlier, this stage called dower. And the dower stage is really interesting because it's kind of a form of suspended animation. You know, I like to refer to it as the animal form of a spore, where they'll enter into the stage and they can hang out in this form for up to 4 months, after which, if they get the appropriate growth cues, they can exit the stage and enter into reproductive development with virtually no consequence, particularly on their reproductive fitness. So when I think of dower, I think of all these great science fiction movies where the astronauts plug themselves into the pods and they go on these long duration missions to Mars and other galaxies. And then they can reanimate themselves once as they get to their final destination. And this is essentially science fiction, but dowers are science reality, and they do basically the same thing. They can live 40 times longer than the normal reproductive animal in terms of lifespan. Now, the things that control entry into this strange suspended animated state include some critical signaling molecules and pathways. And I've listed them here. Insulin-like signaling, we're going to see a lot of this today. DAF2 is the insulin receptor in C. elegans. And we have these extraordinary temperature-sensitive mutants that allow us to make dowers in the laboratory just by switching temperatures. TGF-beta is also involved in the decision to make dower or to do reproductive development, cyclic GMP, and more recently, interleukins are also involved in this division or this decision. Dowers, once they've actually committed to forming or entering into this stage, will radically alter their morphology. And although it doesn't look so radically altered for you, because you're probably not so used to looking at C. elegans, this does not look like a normal C. elegans animal. It's radially constricted. Its buccal opening is plugged, so in fact, it can't eat and doesn't eat over the whole time that it's in dower. It changes its behavior. It no longer cares about food. So you'll find dowers on the top of these agar plates or down the sides, and we talked about the students. You can never find them. They're just doing what you wouldn't expect. They don't care about food. Their metabolism is radically modified. Because they don't eat, they have to live off the resources that they accumulate prior to going into dower. And so they switch a lot of their metabolism around. So they really count on a lot of lipid metabolism and something called the glyoxylate shut. And then, of course, one of the reasons that we're really interested in dowers is because they enter into a state of developmental quiescence, and all the cell divisions become arrested, including the germline stem cells. And over the years, this seems to have been correlated through a lot of different studies with changes in the small RNA populations and chromatin modifications that are very typical to dower. So in order to understand more about the genes, as I said, that might be controlling the ability of this animal to enter into a quiescent stage, we carried out genetic screens in C. elegans to try to find animals that couldn't enter into this quiescent state. In fact, we focused on the germline and tried to find animals that had germline hyperplasia because the animals continued to allow their germline stem cells to divide during this dower state. And we were really lucky to obtain a dominant negative mutation in a really interesting protein kinase called AMP-activated protein kinase, or AMPK. And up until that point, AMPK had been heavily studied for its role in metabolism. It plays a really important role in adjusting metabolic changes in response to low energy within cells. And in a nutshell, it will shut down a number of pathways by phosphorylating key proteins, and in doing so, allowing for a conservation of energy while also phosphorylating other targets to produce energy within cells. Now, the dominant negative mutation that we found disrupted both catalytic subunits of the key protein kinase, which is made up of a heterotrimeric complex of a beta and a gamma subunit shown here. And the catalytic subunits are subject to activation by another important heterotrimeric complex that consists of the tumor suppressor kinase called LKB1, or in C. elegans PAR4, and its auxiliary factors, another factor called STRAD and another called MO25. And together, these two kinases form a signaling axis that we refer to as the LKB1 AMPK signaling axis. And we noticed that in AMPK mutants, so the dominant negative, or later on, as we were able to get mutations in each one of the catalytic subunits, AMPK mutants always had this increased population of germline stem cells, or germ cells, during the dower stage. And more importantly, any mutation that affected any component of the signaling axis was reflected in the hyperplasia that we could quantify in all of these different animals. And what's more exciting is the hyperplasia was a direct readout of how severe the mutation affect the signaling flux that went through that entire pathway. And I've just shown you some of the examples of the mutants that we got in our screens. As you can see, the controls are usually made up of about 32 to 34 germ cells in this DAPI-stained animal shown here. Mutations in the upstream activating kinase, PAR4, give rise to this almost four-fold increased complement of germ cells. This is a beta subunit, STRAD, and the other allele of STRAD that we identified. Now, I think when everybody looks at each one of these germ cells, it's pretty easy to see that with a 3- or 4-fold complement or increase in the complement of germ cells, that with all these extra cells, you could end up with a reproductive advantage if each one of those germ cells was actually competent. And so we're really curious to see if these mutants would somehow have more progeny after they came out of Dower. It turns out, in fact, this is not the case. First of all, if you look at AMPK mutants, and we do almost everything in a DAF2 background, this is an insulin signaling compromised background that allows us to form Dowers at 25 degrees Celsius. But if you raise the animals at 15 degrees Celsius, they don't go through Dower. And as you can see, whether AMPK is present in its wild type form or whether it's completely null, it has no effect on the abilities of animals to give rise to a new generation of progeny. But if you run the same animals through Dower, just by putting them at 25 degrees Celsius, the DAF2 or insulin receptor mutant animals are fully fertile after they come out of the Dower stage, post-Dower adults. But if you lack AMPK during the Dower stage, of course, you get this hyperplasia in the germ line. But once as you recover from the Dower stage and become an adult animal, all of those germ cells are incompetent and the animals are very high penetrantly sterile. So when you see things like that as a geneticist, this high penetrance of sterility, it becomes a beautiful background to do further screening. And so a student in the laboratory carried out a mutagenesis screen where we look for animals that would suppress the sterility in post-Dower adult animals that lack AMPK signaling. I'm going to skip this. And he was able to identify a single mutant called TBC7. And TBC7 is a RAB-GAP protein that's expressed in neurons. And this was really kind of satisfying because in previous work, we identified that we could actually restore a great deal of the AMPK activity, particularly the ability to control those germ cell divisions in Dower animals, simply by adding back the AAK, or the catalytic subunit, in neurons. We first did it just globally expressing AAK2, catalytic subunit, in all of the somatic cells. And you can see that there are 3 lines here that fully restore both the germ cells and also the ability to give rise to progeny in the next generation. So there was some sort of role for AMPK that was in the neurons that was somehow affecting the germ cells. And with this new allele of TBC7, this really confirmed our suspicions that somehow AMPK was interacting with a gene activity in the neurons that had to somehow impinge on decisions to divide within the germline. And we tested this a little further by generating strains that would carry out RNAi in very specific tissues. And we asked the question whether TBC7 was required within the germ cells or within the neurons. And of course, based on this expression and consistent with this, TBC7 was required only within the neurons. And if you eliminated it, then you could actually suppress all the defects that we saw with the AMPK mutants. So there's something going on in the neurons that is impinging on the decisions of the germline stem cells to undergo divisions during the Dower stage. And if you eliminate TBC7 in the neurons, then you can restore the ability to carry out quiescence and also to remain competent in the adult germ. So as I said, TBC7 is a Rab-GAP protein. And the Rab-GAP protein has this classic TBC domain, which is shown here, along with another domain, it's called a TLD domain, which is important for interacting with a Rab protein or the specific Rab proteins. And to our satisfaction, we also noticed that at serine 115 within TBC7, we identified an AMPK phosphorylation sequence that was really fit very nicely to the consensus. So we're excited about testing whether AMPK might phosphorylate TBC7 directly on that serine residue in order to carry out its functions in the neurons to affect the germline. So we carried out 2 series of mutations. Of course, it's typical kinase biology here. We made non-phosphorylable variants by switching the serine to an alanine, which can't be phosphorylated. And in another series of experiments, we switched the serine to an aspartate residue, which would be phosphomimetic. And we examined the ability of those variant animals to give rise to viable embryos. And so when we express a non-phosphorylable variant of TBC7 in a normal DAF2 animal, we notice that, in fact, we reduce the fertility of those post-Dower animals, consistent with it acting or recapitulating or phenocopying what we see with the AMPK mutants. So we interpret this in such a way that AMPK would normally phosphorylate that residue in TBC7 and somehow attenuate its activity. And in doing so, it would allow for the neurons to somehow tell the germ cells that you have to stop dividing so you can be competent later on to give rise to progeny. Now, if that phosphorylation is downstream of AMPK and is actually the business end of the AMPK signaling from the neurons to the germline, then the phosphomimetic variant should completely rescue the AMPK mutant phenotypes in the sterility of those post-Dower animals. And so when we look at those phosphomimetic variants in an AMPK mutant this time, we notice that we can restore fertility in those animals, consistent with AMPK phosphorylating that residue to ensure that TBC7 is somehow attenuated so that the neuronal signals can move from the neurons to the germ cells. And now what we believe, based on this interaction with other TBC, because this isn't the first time that TBCs have been identified as being targets of AMPK, it appears that phosphorylation caused by AMPK, in this case on that residue, gives rise to an autoinhibitory conformational change. So it somehow blocks the ability of TBC7 to inhibit its Rab targets. So let's just recapitulate here. TBC7 is a Rab gap, so it's a negative regulator of Rab biology. And of course, RABs very often are bound to GTP in their active state. And TBC7 acts as a GTPase accelerating protein that will enhance the hydrolysis of GTP so that it goes to its GDP bound state. That would suggest then, in the absence of TBC7, RAB in its GTP bound state promotes fertility in the post-hour animal in AMPK mutants. And if it's consistently in the GDP bound state, the animals will be post-hour sterile. Okay? And so we did a major survey of all the RAB proteins that are predicted in C. elegans genome to identify which RAB might be the target, or at least associating with TBC7. And the one RAB that really stood out was RAB7. And we noticed that if we eliminate RAB7 function using RNAi in an AMPK background, either in the germline or in the neurons, we noticed that when RAB7 was gone in the neurons, you more or less recapitulated this idea that, in fact, you no longer get the appropriate regulation of the germ cells. So RAB7 seems to be the target of TBC7 in the neurons. Its role is in the neurons. It's not in the germ cells. So TBC7 is interacting with RAB7. And when TBC7 is not phosphorylated, it will ensure that RAB7 is always in the GDP bound state. When AMPK becomes activated in these energy stress conditions, TBC7 is blocked, remember, due to that phosphorylation event, and RAB7 will be maintained in its GTP bound state to carry out its function within the neurons to communicate to the germ cells. We've sort of shown that in this cartoon here. But one thing that really comes up is, how is it that this RAB can somehow be communicating between the neurons and the germ cells? Because RAB7 does a lot of things. It's really involved in a lot of critical decisions in endocytic trafficking. One of the key things is that RAB7 associates with the membrane in a late endosome, and amongst other things, it can also give rise to the formation of multivesicular bodies. And so these multivesicular bodies contain these structures called intraluminal vesicles that themselves are associated with RAB7. And under appropriate conditions, these multivesicular bodies, shown here, can move to the plasma membrane, fuse, and then secrete all of those intraluminal vesicles that were in the multivesicular body into the extracellular space, where they can fall into the extracellular space and move to their potential targets. And so we decided that we were going to tag RAB7 with GFP and evaluate whether these intraluminal vesicles that are associated with RAB7 eventually could give rise to these secreted intraluminal vesicles to become exosomes. And so initially, we purified these RAB7 associated potential vesicles. And we've shown that here in this transmission electron microscopy, that a RAB7 molecule, we use an anti-GFP antibody here, is associated with a lipid bilayer encapsulated vesicle, which happens to have about the right diameter or the right size of an exosome. And in addition to that, these purified exosomes, these vesicles with RAB7, also possess all of the critical markers, the criteria that would be typical of secreted exosomes. So we believe then that RAB7 in these neurons is being secreted in the form of an exosome. And perhaps it's that exosome that's providing the information from the neurons to heretically change decisions about cell division in the germ cells. So because we had Rab7 tagged on these particular vesicles, we expressed them uniquely and exclusively in the neurons. And we asked the question whether those Rab7 GFP marked structures could leave the neurons and eventually interact with the germ cells. And what I'm showing you here is an animal where the Rab7 GFP is expressed in the neurons, and yet we detect the GFP signal from Rab7 in the germ cells. And this is really hard to convince a lot of people of. We showed that, in fact, there's no mRNA for Rab7 in the germ cells. However, some people have identified protein for Rab7 there, consistent with that movement from one cell type to the germ cells. We carried out RNAi specifically in the germ cells to eliminate any possibility of an mRNA that encodes Rab7 in the germ cells, and yet we still see the Rab7 GFP protein. We performed experiments with T2A skippers where we only express the proteins in the germ cells. And MK, under this particular marker, RGEF, neuron specific, or this promoter neuron specific, will only stay in the germ cells. Yet we see the secondary gene after the skipper, we see that the GFP from Rab7 still goes to the germ cells. So we're convinced, and I hope I persuaded you, that Rab7 vesicles are made in the neurons and they make their way to the germ cells in a very specific manner. They don't go to other cell types. They go specifically to the germ cells. And I think what's even more important is if we drive Rab7 GFP in other cells different than the neurons, for example, in the intestine or the muscle or the hypodermis, they do not go to the germ cells. There's something important about those structures that are made in the neurons that tells them to home to the germ cells with that information. Now, what is this information? Because exosomes have been shown to carry a very heterogeneous cargo. It could be anything from proteins, it could be cytokines, it can be DNA, it could be mRNAs, or it could be microRNAs. They're all there within exosomes, and everybody's seen something that they like within these particular vesicles. But our first hint at what might be important for the signal came from studies that were done completely independently in a collaboration that we had started with Eric Miska in Cambridge. And Eric was able to show that in AMPK mutant animals, in the dowers, that the microRNA population was dramatically suppressed. And as an aside, this is completely different than all the other small RNA populations, which, according to him, he'd never seen such a radical increase in all the other types of small RNAs. But nevertheless, the microRNA population was suppressed. So that started to tell us something, that maybe it was these microRNAs that were critical for sending that proquiescent signal to the germ cells from the neurons. And that might be loaded into this particular Rab7 associated cargo. And further evidence came from this when a student in my laboratory decided to take, and this was, again, a heretical experiment. He decided to take total RNA extracts from dour animals that were made from this insulin receptor mutant and from AMPK mutant animals. This is just total RNA purified from both of those strains. And he mixed that RNA with AMPK mutant larvae and just allowed them to take it up. And he was able to rescue the sterility defect just with a total RNA extract. And he couldn't do the same thing with the extract that he purified from an AMPK null mutant animal. And once more, I'm not going to show you the data, but I can share it anytime you like. He did a poly-T column on those RNA extracts to separate mRNA from other types of RNA. And the poly-T bound fraction had no effect on the sterility of these animals. It was in the flow through those columns. So it's not an mRNA. And probably the coolest experiment is he took all those RNA extracts and treated them with RNase A at either no salt or 0.5 molar NaCl. And at 0.5 molar NaCl, the RNase A will chew up single-stranded RNAs, but they'll leave double-stranded RNAs. And with that 0.5 molar NaCl fraction, he's able to rescue the defect in sterility in these animals. All of which points to some non-mRNA, RNA-based rescuing factor that is presumably in a double-stranded state. And with this information from Eric, we started to think that, in fact, this has to be microRNAs. And strangely enough, I promised Victor that I would never work on microRNAs, but I think this alludes to something that's true for all of us. No matter how hard you try to hide from microRNAs, they're going to be involved in the pathway that you're interested in. And so we tested a number of components that were involved in all the other small RNA pathways for their ability to somehow compromise the sterility or the fertility in these animals. And we found that dicer-1 was very important, and so was DROSHA and PASHA. DROSHA and PASHA are two very important proteins that are required for the appropriate biogenesis of microRNAs. So we have dicer, which is responsible for a lot of small RNAs, but more importantly, DROSHA and PASHA that really point the finger at microRNAs being involved in these decisions. And we look at this even closer by using our tissue-specific RNAi strains. We notice that, in fact, you absolutely require dicer and DROSHA and PASHA in the neurons, not in the germ cells, in order to contribute to this regulation that's taking place between the neurons and presumably the germ cells. So I think we're pretty convinced, at least genetically, that the microRNA biogenesis pathway is somehow involved here. And presumably, it may be that these microRNAs have to be made within the neurons and somehow loaded into vesicles that contain Rab7 that leave the neurons and eventually impact decisions in the germline. So to test that possibility, we designed a sensor. And the sensor consisted of a histone GFP reporter that had some microRNA binding sites, seed sites that could be targeted by a specific microRNA in the 3' UTR. And we drove that sensor exclusively in the germline using germline-specific promoters. And that's shown here. And in the neurons, we decided to use a promoter that we've been using throughout, RGEF, that's very exclusive to the neurons, but we're driving miR51. And miR51 is a microRNA that is highly repressed in our population of decreased microRNAs. It's involved in a lot of cell growth, cell division genes and repressing those. So we thought that would be a really good procreation signal. But in addition, we had miR51 mutants so that we could introduce miR51 in the neurons as the only source of miR51, this microRNA in the whole animal. And then we tested whether miR51 is produced in the neurons can somehow affect this sensor that's only produced in the germ cells. So if the microRNA activity is high, that's to say miR51 is being produced in the neurons and makes its way to the germ cells, and it attains its targets, it should extinguish the GFP. So the GFP should be low. By the same token, if the microRNA activity is very low, then there should be high GFP activity in the sensor in the germ cells. Okay, are we on board with that? And so we tested this. Now, this is just a control to show that in adult animals, the sensor is present in all the germ cells, and we can see the GFP. And there's no difference between just dour animals and dour animals that lack GFP or that lack AMPK. And that's because they're not being challenged. They were never put through dour per se. These are just adult animals. They never, ever had to see dour develop. But when you switch those animals to 25 degrees Celsius and you put them through dour, we notice that there's a dramatic drop in the GFP, consistent with very active microRNA activity, right? That's how our sensors work. The miR51 that's being produced in the neurons is somehow making its way to the germ cells, and it's extinguishing its targets, okay? And if we eliminate AMPK in those dour animals now, suddenly we reverse this effect. The miR51 can no longer make its way to the germ cells, and the sensor is on really quite strongly. That's quantified for you, so you don't have to really examine all the GFP producing cells. But it's really quite dramatic. So by eliminating AMPK in these animals, you somehow disrupt the signaling between the miR51 that's being produced in the neurons and the target that was normally extinguished in the germ cells. Now, the next question is this taking place through a Rab7 dependent mechanism whereby potentially these Rab7 associated vesicles will take that microRNA and potentially bring it to the germ cells to then provide the germ cells with the mRNA through this exosome-mediated communication. And to do this, we carried out Rab7 RNAis and tested to see whether indeed we could eliminate that particular sensor signal, the GFP. And as you can see here, that the GFP remains really quite strong in these animals that produce the microRNA in the neurons, but for whatever reason, you don't have Rab7, you can't get to the germ cells to extinguish the GFP that's associated with the target, all right? So you need Rab7 in order for that microRNA to leave the neurons and to attain and extinguish its targets in the germ cells. And we've quantified that here. Now, one of the questions I get from all my small RNA colleagues is whether or not the microRNAs are naked when they go into these potential vesicles and leave the neurons, or whether they're already preloaded with their associated Argonaute proteins and the RISC complex before they get encapsulated into these vesicles and leave to carry out their functions. And so we sought to figure this out by, again, driving Argonautes, in this case, ALG1. It's a specific microRNA-associated Argonaute. There's 3 Argonautes that are really important for microRNA biology in C. elegans, and ALG1 is the predominant one. So we labeled ALG1 with MK, drove it using our exclusive neuronal promoter, and we evaluated, just like we did with Rab7, whether we could detect ALG1 within the germ cells. And of course, I wouldn't be here telling you about it if in fact that's what we saw. So what that tells us then is that ALG1 is associating presumably with those vesicles, the Rab7 associated vesicles, and presumably they're making their way to the germ cells in a similar manner in this Rab7 dependent manner. And of course, to test this, we eliminate Rab7 using RNAi, and we show that in fact, in order for ALG1 to get to those germ cells from the neurons, you require Rab7. Consistent, again, with these vesicles transporting not only microRNAs, but their associated risk assembly in a vesicle that eventually homes to the germ cells in order to provide a proquiescent signal to the germ cells to tell them to stop dividing. And of course, anybody can argue that, yeah, okay, maybe that's true, but maybe the Argonaute proteins are just associating non-specifically with the exterior of the vesicles. And that's true for a number of other types of vesicles. It's not on the inside, but it's on the outside. And we tested this biochemically by carrying out proteinase K experiments on the purified vesicles. And if you carry out a proteinase K experiment on vesicles in the absence of a detergent, you can't degrade the Argonaute proteins, suggesting then that they're protected from that protease while they're in that vesicle environment. But if you pre-treat those vesicles with a detergent, Triton X100, of course, and then treat them with proteinase K, you eliminate the Argonaute proteins. And that is pretty strong biochemical evidence that the ALG proteins are on the inside and they're protected by that lipid bilayer. So we don't know how those microRNAs are selected. Presumably they're all, or they have this proquiescent signal that they're sending to the germ cells. I don't know if there's a specific transcription factor that drives the expression in neurons, or whether there's just an existing pool of microRNAs all over these cells, and then somehow they get selected to go into these particular Rab7 exosomes. But of course, they're going to eventually be associated with the risk, and then at some point, that risk mRNA or microRNA complex has to be incorporated into this exosome. And how that takes place was really curious to us, and we were really trying to figure it out for some time until this beautiful paper by somebody from Ron Kahn's lab, I assume it's a postdoc, Garcia Martin, that wrote this paper where he described how tetranucleotide motifs on microRNAs actually help the cell to decide whether a microRNA stays within the cell per se in the cytoplasm, or whether it's going to be loaded into an exosome and secreted for its function somewhere else. And these tetranucleotide sequences, he nicely referred to them as exomotifs. And we started to look at the presence of exomotifs on a lot of those microRNAs that were depressed in our data set that came from Eric Miska. And many of the microRNAs that he identified had these exomotifs, sometimes one copy, sometimes two, and sometimes three copies of these exomotifs. And so we're curious whether the exomotif played any important role in the loading of these exosomes and the overall function of these Rab7 associated exosomes. And so luckily, on the miR51 microRNA that we've been using, we notice that there's a very nice exomotif underlined here and indicated. And we decided to scramble that exomotif, but also maintain the purine-permitting ratios. And we called that our scrambled exomotif, for want of a more creative title. And then we tested whether that scrambled exomotif could behave like the wild type miR51 in our sensors. And when we express the scrambled variant of the miR51 microRNA in neurons and test for its ability to extinguish the GFP in the sensor that's expressed in the germ cells, we see this completely ineffective. So that exomotif is critical, at least for attaining and extinguishing the GFP targets mediated by the ability of the microRNA to interact with its seed sequences. And I have to add here that based on computational analysis anyways, the change that we made here has no effect on at least the thermodynamics of the binding of the microRNA to its targets. So we really believe that the exon motif isn't affecting the ability of microRNA to bind to its seed sequences, but rather it's affecting the ability of the cell to recognize that microRNA as something that has to be loaded into the exosome in order to be secreted and eventually make its way to the germ cell. So one of the big mysteries, now that we have a pretty good idea of what might be going on in the neuron and this communication between the neuron and the germ cells, what we really don't understand is what happens when the exosomes get there and how the exosomes know that they actually arrived at the final destination. But we started to look at proteins that have been examined in the past that are really critical for this mobile nature of RNA. And a lot of this work really is downstream of Craig Hunter's work, and probably even more notably, Anthony José's work. So Anthony José some time ago identified this possibility that microRNAs and small RNAs might be moving around in the animal in what he calls this mobile RNA. And we looked at a few of the genes that he identified as being really critical for some of this mobility. And we tested them for their role in our particular system. And we know that if we eliminate SID3, which is this really important protein for systemic RNAi, but it tends to be important for uptake of these small RNAs, we noticed that SID3 is absolutely required in these mutants that we've been examining in the germ cells. It's one of the only ones we've identified so far is required in the germ cells in order to somehow transduce this proquiescent signal to the germ cells so that the animals will arrest their cell division and eventually remain competent post-hour. On the other hand, if we look at other SID proteins like SID5 and a few others we tested, SID5 is not at all required in the germline. It's required in the neurons. And SID5 plays an important role in endosomal trafficking, which is consistent with probably the maturation of the initial exosome to start with. And the same is true for receptor-mediated endocytosis 8 or RME8, which again is required for appropriate maturation in the endosomal trafficking pathways. It's required in the neurons and not the germ cells. And so because we're really interested in how these exosomes might be taken up by the germ cells, we decided to see what would happen if we tested our sensor in animals where we eliminated SID3 activity by RNAi. And of course, if you eliminate the SID3 activity, we find that, in fact, the sensor can't be extinguished, consistent with it playing a role at the very end in the germ cells and somehow taking up those exosomes, presumably, and liberating their proquiescent signals so that they can actually attenuate some of these genes that are important for divisions in the germ cells. And we did the same kind of experiment, except not this time using RNAi. Anthony José showed that SID3 is a tyrosine kinase, very important for endocytosis. And if you eliminate the kinase activity, you more or less disrupt this endocytic process per se. And so we drove a kinase dead variant of SID3 in animals, and we're basically able to reproduce the same kind of thing, whereby the GFP sensor that's targeted by miR-15, this classic sensor I've been describing to you for the last 10 slides, can no longer be extinguished. So it relies on this ability of SID3 to endocytose presumably these microRNAs from where they're produced in the neurons so that they can get into the germ cells and eventually couple with their targets and extinguish their expression. And that's been quantified over here. So what I described to you today, and I hope I convinced you of, is that AMPK is playing a really important role in neurons and not in the germ cells like we initially thought. AMPK is critical for the first step in attenuating the activity of a Rab-GAP protein within specific neurons. And by blocking that activity, by phosphorylating it, it allows for Rab7 to somehow develop or form or establish these exosomes that will leave the neurons into the extracellular space and then home to the germ cells in a very specific manner. And once they arrive at their destination, somehow SID3 is going to allow for those exosomes to enter into the germ cells, presumably free their microRNAs so the microRNAs can interact with their targets, thereby telling the germ cells to stop their divisions and to hunker down for the long haul. And what I think that we show here is that, in fact, there's a very important role for small RNAs and this communication via these exosomes between one tissue and another. And I think initially we thought that everything was happening in the germ cells, but now I'm really convinced that it makes sense that you have designated neurons that are very sensitive to these energy changes. And those changes in the neurons are critical so that you never let your critical cells, these germline stem cells, enter into a stage of energy deprivation. And so that you can make all of those changes to enter into a quiescent stage before they ever see anything like running into AMPK deficiencies. And so with all that, I have to thank two of the graduate students that did most of it. Christopher Wong did the bulk of the work on TBC7 and the exosome work. He's now working, not surprisingly, with Eli Lilly in their RNA therapeutics facility in Boston. And now Lena Jersak, she's still in the lab. She's doing some really cool stuff with a lot of the other microRNA, small RNA switches that are mediated by Dicer. Pratik and Shaolin, these have always been great collaborators. Eric Miska was really essential for some of our hypothesis generating data. Fabian Brockman, who did the work in his lab. And that's Chris and Elena. And thank you once more for inviting me. And I'd be very happy to take any questions that you have one-on-one. Yeah, that's a great question. So I can't say much about the anatomy. The person, if you want to talk about anatomy, it's Claire. But what I can say is that, and I didn't show you the data now because I'm already getting pretty late, is that we actually narrowed it down to serotonergic neurons. So initially we're doing everything with this pan-neuronal promoter. So all the neurons express RGAF, right? And one of the reviewers of our paper, and rightfully so, was very curious with the same kind of question. If you're expressing, these exosomes far away in a distal cell, it doesn't make much sense that it can make its way all the way along and find the gonad, right? Turns out that it looks like this is very specific to serotonergic neurons, and I'd even go as far as to say now we're pretty clear on it, that this is happening in the HSN neurons, which are just next door. So that makes a pretty good case for them just being secreted into the extracellular space, and it's like, look who's next door kind of thing, right? And they go to the gonad. So I can't say about the entire repertoire of targeted genes. I can tell you that many of the genes that are misregulated, the mRNAs, we do RNA-seq in these animals, many of the mRNAs that seem to be misregulating AMPK mutant hours have to do with growth and division. And we've looked at the 3' UTRs of a lot of those, and of course, it helps us to go back and figure out which are the critical microRNAs. And that's kind of why we came back to miR-51, because it seems to target a lot of them. But there's no one single microRNA that targets the whole repertoire, right? There seems to be a number of other microRNAs probably that are important to hit all of these mRNAs that seem to be misregulated. Having said that, if you drive like miR-51, it's surprising the kind of rescue that you get. So it looks like if you pick the right microRNA, you can correct the hyperplasia in the germ cells, and also like the competence, the fertility as well. So miR-51 is a big one. I think miR-34 is another big one. These are classic growth affecting microRNAs. Highly conserved. A great question. Yes. Yes. Yeah, that's a great question. It's a really great observation. Fact is, I don't really know. Honestly, I think it's a different pathway. I think the microRNAs are somehow getting in and doing their job to the germ cells, but it may be another pathway. And why I say that is, so there's this really great small RNA biologist at Pasteur who presented at the RNA meeting last year. And he basically showed that I think that there's this beautiful, I guess, dialogue between the gut and the germ cells, and that he was somehow getting microRNAs that are expressed in the gut into the germ cells, through a pathway that I don't think is necessarily shared, but may be relevant in the future. We just don't know. It may have to do with exosomes as well, but they may be a little different. It might not be the same Rab7 associated exosome. But clearly, he was showing that microRNAs are moving from the gut to the germ cells in various challenges. In our case, they may be packaged. It's certainly not the same way that they're coming from the neurons, but I think that it has much more to do with this gut and germline relationship. So no, I don't know that. No, not yet. I've been bugging people to like screen, to just find out what's involved in this pathway, and we could figure that out. But I don't have so many hands anymore. But it's a great question. It's an intriguing. He did tell me he was going to do this experiment. I'm on the record for saying this is garbage. It's not going to work, right? And he did it, and I just, it's one of those classic supervisor moments where you just go, my God, biology is so great. It's surprising sometimes. But initially, I wasn't sure, but you can do all these manipulations. And the salt experiment with RNase A was the killer for me. That you get rescue with that. It's really easy. So I'm going to go up to Jeremy and I come back here. Is that okay? Yeah. Yeah, so that's a great question. I don't know is the answer for the other cell types. I don't know that they necessarily make Rab7 associated exosomes that would leave per se, because on those exosomes is probably a whole lot of other proteins that may be, in this case, serotonergic neuron specific. And maybe, for example, in the intestine, Rab7 is just doing its good old job in maturing the late endosome and deciding whether it's going to go to the the lysosome or do something else, but it might not necessarily secrete. So it's the ability to form these multivesiculated bodies and the intraluminal vesicles that seems to be critical. And I don't know what is involved in that decision. All I can say is that we don't see the Rab7 leaving those cells, going to another cell type. because we can only see the Rab7 where we expressed it. And it certainly doesn't go to the germ cells. So I really don't know what's going on. That's not to say that there aren't other exosomes that might be doing similar things in different cell types, but might not be Rab7 associated. And I think, I alluded to a few people, I'm more excited these days about looking at things like Rab10 or other Rabs that may direct the same kind of event, but it might not be the AMPK dependent. We're just lucky because we got to use genetics to really figure out this pathway. But there may be other exosome-driven intertissue communication phenomena that exist out there that don't have anything to Rab7, but rely on Rab10, Rab11, or some other Rabs. Does that answer it? Now here, yeah, sorry. So this is pretty specific to the Dower stage, but it doesn't, okay, and this is me hand waving here. I can't believe that something so elaborate would just be set up for the Dower stage. I think that when you have this sort of devoted machinery to secretion and uptake, and the way that these microRNAs are working, I can't believe that this is just uniquely for Dower's to send out a procreation signal. And it's really up to us to figure out the other pathways that might be in a regulated kind of fashion. So okay, well, I think I can tell you, like if Eric, who did the small RNA, analysis, Eric really looked at everything during Dower and he looked at post-Dower. And when, in a nutshell, if we look at Dower animals that lack AMPK signaling, they turn on genes that are typical of a normal post-Dower adult animal. So they don't even recognize that they're in Dower. Despite the fact they look like Dower, they behave like Dower, they do a lot of things that, Dower specific. And I think that's because they're very separate pathways associated with what a Dower is, right? But on the inside, metabolically, and all the gene expression suggests that these animals think that they're adults and they're doing all the kinds of things that adults do, but it's miscoordinated in a developmental chronological fashion, right? And so they screw it all up because they do it too early. They make sperm during Dower. Like this is crazy. You almost never see his phenotype. But AMPK mutants, they'll make sperm like it's going out of town. And that's something that only happens in the L4 stage. So it's I don't want to use heterochronic loosely because of where I came from, but there's weird things with timing. It's all screwed up. So in the end, during the L3 under normal circumstances, when growing conditions are good, the germ cells proliferate like crazy because they're starting to make all the gametes, preparing to make the sperm, and they're starting to make all the gametes. And they're doing the same thing in the Dower animals that lack AMPK because those constraints are lost. So they're really doing what they do normally in an L3 or an L4 animal. Yeah, but they really screw up, boy. Anna. You know what? So yeah. So I'm holding back on that conclusion right now, not because I know and I don't want to share it, it's because I don't know. But I'm really holding back on that. So we're purifying these things, and we've learned it's taken us some time, and it's hard because it's like one class of neurons that make them really well. So we're purifying them now, and once as I get the microRNA sequences, and I'm confident about what we're seeing regularly in these things, then we'll start to talk about what the targets are how the implications might be involved in other diseases. But for now, I think it's just hand waving. We don't know exactly what the targets are until we actually get the microRNAs. That's how I feel anyways. I'm not that conservative, but pretty conservative.